Good morning and welcome. My name is Will Gray-Roncal, and I'm an organizer of this symposium, along with my friend and colleague, Brock Wester, from the Johns Hopkins University Applied Physics Laboratory. I want to introduce our topic today, entitled Toward Petascale and Exascale Connectomics. And with the advance of technology and collaboration between engineers and neuroscientists, it's now possible to estimate networks of the brain at the single synapse resolution. And so we're excited today to bring together speakers to present recent research breakthroughs and their perspectives on the field, as well as related analyses that may inspire future research. So collectively in the field of connectomics, we work to image brain tissue and estimate brain networks to answer hypotheses about the brain's structure and function, converting the raw biological tissue into a format that can be more easily analyzed by neuroscientists, computers, and mathematical techniques. Even in the early days of neuroscience, of course, we were able to capture information about individual neurons, and more recent technology advances have allowed us to look at the brain through many different lenses and technologies. However, microscale circuits are still not as well understood and require advances in scalability, scientific understanding, and community engagement. To interrogate the brain, we have many different options, ranging from magnetic resonance imaging on the left, where a cubic millimeter is represented by a single byte of information, to the electron microscopy data we focus on here, where each pixel is a few hundred cubic nanometers, and a cubic millimeter is 2 petabytes. Depending on the approach taken, we can then make brain graphs at different resolutions, ranging from putative connections between brain regions to detailed synaptic level connections between individual neurons. We think this work's really important because understanding these microcircuits is a missing link to understanding many neurological diseases and informing diagnosis, prognosis, and treatment. We think these models will have a great impact on diseases and conditions such as traumatic brain injury, autism, and Parkinson's disease. Also, this field may be a key enabler for replicating brain function and understanding and validating theories of neural computation and understanding the brain. Here's an outline of some of the steps commonly taken in connectomics discovery. Our speakers will touch on advances, ideas, challenges, and solutions in many of these areas, from thoughtful experimental design, imaging, data collection and storage, segmentation and meshing, proofreading, and analysis. It's an exciting time to be in connectomics, where we're really at this inflection point in technology, where many of the classical challenges in scalability and data generation and analysis now are achievable through state-of-the-art techniques. Our lab's focused on solutions in scalable storage, evaluating the quality of these large-scale circuit reconstructions as they're developed, mining that information for motifs, for computational primitives and small networks, and then applying what we've learned there to novel computational models. And I think you'll hear from many labs here and around this meeting for others who are doing similar work. It's a really exciting time, and there's much still to do.
The field of connectomics is rapidly accelerating, and as data sets continue to grow in size, we are able to leverage technology to target much larger questions. In just a few years, we've progressed from a brute force reconstruction of a C. elegans worm to contemporary efforts to reconstruct a cubic millimeter of cortex or a Drosophila brain. Networks of an entire mouse and large-scale comparative network questions are now within reach. We're excited to share this progress and some of the emerging research that will catalyze the future of this field.
Here today, we've gathered emerging and established leaders in the field of connectomics and are excited to talk with you today in this new and innovative virtual format. And I'll leave you with a quote from the founder of neuroscience, Santiago Ramon y Cajal, who says that the brain's microcircuits are impenetrable jungles where many investigators have lost themselves. And I think it's a really exciting time, like I mentioned, where we're uniquely poised to help understand the structural and function of the brain. And so thanks to everyone who made the symposium possible, for all of our speakers. And first up is Jeff Lichtman from Harvard University.
Connectomics began with the work of Sidney Brenner, a molecular biologist, and his colleagues John White, Southgate, Thompson, who published after 10 years of work the structure of the nervous system of C. elegans, 40-page paper that itemized the connections of every one of those 300 cells. That's the past of this field. If we fast forward 40 years to now, the same kind of reconstructions can be done so much quicker. Here's a paper that was just put in bioRxiv. It is 8 connectomes, including 2 adult connectomes of C. elegans, all done in a 15-page paper with the wiring diagram of every cell. And instead of requiring 300 pages, it's 15 pages because all the data is now online. If this is the present, what is the future? And what about, instead of the mind of the worm, what about the mind of a mouse? Could one do an entire mouse? Let's talk about this just for a second. A mouse is much bigger than the 300 neurons or so in C. elegans that work. You have about 100 million neurons, 100 billion synapses, and the volume is exceptionally large. It's 500 cubic millimeters. That would be equivalent to a box that's approximately 8 by 8 by 8 cubic millimeters. And that's worth saying because the state of the art right now is 1 cubic millimeter. So this would be a 500-fold increase from where we are right now. Not only is it a large volume that would have to be imaged, but it would be a very large data set. Depending on the voxel size, which would be somewhere between 500 cubic nanometers to maybe 1,000 cubic nanometers per voxel, this data set would be somewhere between 500,000 and 1 million terabytes. 1 million terabytes is an exabyte, and that would be a tremendous challenge, obviously. To give you a sense of why the mouse is sort of an attractive animal to imagine doing a complete mammalian connectome on, in a logarithmic way, it is sort of halfway between the most completely studied nervous system by connectomics, which is the fly brain, which has about 100,000 cells. So you multiply that by 1,000, you get to the number of cells in a mouse brain, 100 million. And 100 million is 1,000-fold smaller than the number of cells in a human brain, which is 100 billion. So you'd need about an exabyte of storage. No one wants to do this for multiple generations. It would be nice to be able to complete this in even half a decade, at least the image acquisition part. And that is possible if one parallelizes the imaging using machines like multi-beam scanning electron microscopes or other fast methods and having 20 or 30 machines. It would be expensive. Just the cost of buying all those microscopes would be $100 million. So you would have to multiply that number by several, maybe a small number of integers to get to the cost of the whole project. So this won't be an easy project if it goes through at all. There are technical challenges that have not yet been solved, and I would like to just list them very quickly. One is metallizing a whole mouse brain. Getting osmium through a whole brain is going to require materials scientists and organometallic chemists thinking a little bit more seriously about how one can get a metal that is highly reactive. Osmium tetroxide is highly reactive all the way through a brain. Secondly, you have to section and image something that is much bigger than the volumes that have been done so far. This will probably require physicists and mechanical engineers to be thinking about the most efficient ways of doing that. One thing that has to happen is it has to be very robust. The sectioning and imaging can't lose anything. If you even lost 200 nanometers somewhere in the middle of the volume, many processes would be lost. Similarly, the throughput has to be very high. I estimate maybe in the range of about 1,000 terabytes per day in order to finish a project like this in half a decade. And finally, you'd have to deal with the scaling up of this huge number of computational steps that have made connectomics work so well. And computer scientists, and especially those interested in deep learning and artificial intelligence will have to be playing a role. All of stitching and alignment has to be done between sections by computer scientists. Segmentation has become really quite powerful, but doing it at the scale is just a new challenge. And one would have to build analysis and rendering tools to deal with an exabyte of data. And finally, there has to be a way that this data can be easily shared with as many people who want to look at it as possible. None of these things are trivial to do. So in conclusion, I'd just like to ask the question that maybe some people are wondering about is why might we want to do this? And I'll just give you a quick bunch of things: a path towards unbiased catalog of all the cells and synaptic connections of the whole brain with broadly subserved mammalian neuroscience, a path for bridging the divide between non-invasive mesoscale imaging technologies like functional magnetic resonance imaging and ground truth microstructure. How do the synaptic connections relate to the projections? And seeing that in the same animal will be tremendously valuable. This will also be a path, perhaps, for learning something about the structure of long-term memory. Mammals have a lot of behavioral repertoires that come about through experience that are stored stably for the life of the animal, including human beings. What is the structural basis of that? This will also be a path towards describing the neuropathology of brain disorders. Some brain disorders almost certainly will be connectopathies, pathologies of connections. Animal models of disease will be possible. Once one builds this infrastructure, it will be possible to look at diseased animals. This will also be, for computer science, a path towards designing non-biological thinking systems that have more properties that are similar to those of actual thinking animals. For example, many machines can play chess or do other board games, or drive a car, but they can't do many things. A blueprint for general intelligence is found in the wiring diagram of a mouse. Also, we do a lot of few-shot learning as human beings, and other animals do the same thing. We don't need thousands and thousands of examples like our machine learning training data sets, but we have to learn better how that is done. And finally, it would take megawatts of power to build a computer as powerful as a mouse's brain. So we would learn something, I think, about how energy-efficient computing works. In our view, a project like this could likely be a turning point in the human pursuit of understanding what makes mammals, and especially us, the unique animals that we are. Thank you very much. The next person talking is Alyssa Wilson, Princeton University.
Hi, everyone. My name is Alyssa Wilson, and I'm a postdoctoral fellow at the Princeton Neuroscience Institute in Sebastian Sung's lab. And today I'd like to talk to you all about some insights that we hope to get from serial section electron microscopy, or EM, of mouse brain in particular, as we scale this technique up to larger image volumes. That's somewhat separate from the primary goal of this technique that we often put forth, which is identifying neural circuits. And what I'd like to talk about specifically is structural correlates of synaptic plasticity that we can use to learn mechanisms of neuronal rewiring. And so we can get a better sense of what I mean by that. I'd like to just start by reminding us all what electron microscopy reveals in biological tissue. The mode of contrast is metal stain that targets lipids and some proteins. And so in an electron micrograph, what we see is all membrane-bound objects as well as some of those proteins. And if we look at an electron microscopy image, this is an adult mouse in the visual cortex area V1. You can see that it's very densely packed. And the membrane-bound objects involve parts of cells as well as pieces of neurites. I messed this up. If we look at an example of an EM image, this is mouse visual cortex V1, layer 2-3. You can see that the image is very packed with membrane-bound objects, and this includes cells and pieces of cells that we call neurites, as well as some intracellular organelles, like this mitochondrion and an endoplasmic reticulum that I've shown in red and blue, respectively. And if we scale this technique up to take series of images of a piece of tissue, we then get a very powerful set of data, which is the 3D context for all of this that allows us to make sense of it. So if we look at the data set that image came from, this is the proof of principle data set from the IARPA microns contract, and it was acquired by the Allen Institute for Brain Science. You can see that we have quite a few large portions of cells contained within this image volume, including the cell here, which is a neuron, and it has axons and dendrites visible, and the morphology allows us also to assign cell types. This is a pyramidal neuron from layer 2-3. And because we can see the organelles inside of all of these cells, we can also determine where the synapses are based on features like synaptic vesicles and postsynaptic density. And so all of that allows us to see the synaptic connectivity between cells contained in this volume, like the example I'm showing here of a basket cell neuron in blue and a pyramidal neuron in red. And we've been able to detect using a neural network all of the synapses formed by that pyramidal neuron onto the basket cell. So with high throughput electron microscopy, we can start to answer the following questions. First, the question that often we ask, which is how are neurons synaptically connected to each other? But also we can ask, how are intracellular structures like organelles and protein structures related to neural circuitry? Specifically, we can ask, how do these things vary in relation to synaptic strength? And from that information, we can move forward and start to ask, what role might these things have in synaptic plasticity or developmental rewiring? And how can we get that information given the fact that EM gives us a static snapshot of a piece of brain tissue? Well, if we look at an adult that's been through developmental rewiring, we can see by measuring the structural correlates of synapse and organelle function, which synapses are strong and what the differences in organelle features might mean relative to that strength. So to give a concrete example of what I mean by that statement, I want to share some preliminary work I've done on this EM volume on a particular organelle that many people are familiar with, which is mitochondria. Mitochondria are responsible for generating ATP in cells, which is an energy source that's necessary for many cell functions. And also they have another function, which is to remove calcium ions from their external environment. And they do this through membrane-based uptake. And importantly, that means that the size of a mitochondrion is proportional to how effectively it can remove calcium ions. And this is in particular important for neurons, where the calcium ion concentration is tightly controlled, and small changes make big differences in synaptic firing and other activity-related processes. And recently it's been found that at synapses, mitochondria actually halt due to elevated calcium ion concentration. And while they're at those synapses, they produce energy that is required in order to maintain synaptic proteins. And also they affect the amount of calcium ion available for synaptic firing. So this information already tells us that mitochondria might be important for determining synaptic strength or affecting it. Moreover, a few years ago, people found that mitochondria are motile during development when often synapses are adjusted in terms of strength based on activity-dependent experience, but they become stationary by adulthood. So all of this information taken together leads us to ask the question, is there a relationship between mitochondria and the synapse strengths that are established during development? And so since in our proof of principle volume we had a young adult mouse brain tissue, we decided to analyze the mitochondria in that tissue to start and try and get at this question. And so using a neural network that was aimed to detect mitochondria, it's similar to the network we use to detect our synapses, we were able to generate segmentations of the mitochondria in this image volume with a relatively high precision and recall. And overlapping those mitochondria with our segments of the cells themselves in this volume, we were able to get a data set in which every segmented cell, like this pyramidal neuron shown, has mitochondria associated with it that we can then study. So if we zoom in on the cell body of this neuron, you can see every different color is a different mitochondria, and this gives us a lot of information about where the mitochondria are, how they're shaped, and what their surface area and size might be, and that tells us about how well they can, for instance, remove calcium from their external environment and give us a lot more information about what might be going on in the cell. So in this data set, we looked at excitatory synapses and the mitochondria near those synapses. And by isolating the excitatory synapses between pyramidal cells in the volume, we found about 5,000 mitochondria within about 5 microns of synapses. And there were about 1,900 synapses that are formed between the 334 pyramidal neurons with somas contained in this image volume. And just looking at these populations of mitochondria has already given us some interesting information, and I'll share with you some of those preliminary results now. So looking at presynaptic mitochondria, we found that only about 60% of the synapses from excitatory pyramidal neurons onto other pyramidal neurons have presynaptic mitochondria nearby. And if we assess one structural correlate of synaptic strength, that is the dendritic spine volume, larger spine volumes correspond to supposed stronger synaptic strength. And we compare for synapses that do and don't have presynaptic mitochondria, we find an interesting result. In blue in this graph, you see a histogram of spine volumes for synapses that do have nearby presynaptic mitochondria. And in orange, you see the distribution of spine volumes for those that don't have presynaptic mitochondria. And we find that if a synapse has a large spine volume, it likely has a nearby presynaptic mitochondrion. And so this starts to suggest a biological insight, which is that presynaptic mitochondria may have a role in permitting stronger synapses between excitatory neurons in the visual cortex. If we look at the postsynaptic mitochondria in this population we're studying, we see another interesting result. So almost all synapses have postsynaptic mitochondria within 5 microns of the synapse. So if we just look at that proxy for synaptic strength, dendritic spine volume again, and we look at the size of those postsynaptic mitochondria for each of these synapses as a function of spine volume, we see that small spine volumes have exceptionally long postsynaptic mitochondria in some case. So here's the data for that. On each data point here is a synapse in our data set, and the x-axis is the spine volume of that synapse, and the y-axis is the average mitochondrial length. And we see that the smallest spine volumes have very long mitochondrial lengths in their postsynaptic mitochondria. Furthermore, earlier this year, people in our lab posted some work on bioRxiv that assessed the spine volumes in this exact data set, and we found that spine volumes, the distribution is shown in black and it's modeled in red. They're well described by a sum of Gaussians, and we posit that those two Gaussians represent two states. The Gaussian distribution on the left represents potentially a weakened state where spines have smaller volumes, and the Gaussian on the right represents a potentially strengthened state of synapse strength where the spines have slightly larger volumes. And the natural split between those two potential states occurs at about 0.09 microns cubed. And so if we look at that particular spine volume on our data with mitochondria, we find that it's, to a first order of approximation, a good split between spines that are very small and have very long mitochondria and those that are bigger and less likely to have very long mitochondria postsynaptically. So this tells us that mitochondria may have a role in determining spine strength postsynaptically as well. A suggestion is that excessive presence of postsynaptic mitochondria might be involved in synaptic depression or downscaling, and I'm happy to talk more offline about that if people are interested in our thoughts on it. But these two examples give us a picture to summarize of what we have to gain by looking at organelles in these volumes in the future. Visualization of organelles within neural circuits can lead to really important insights potentially about circuit development or learning-based changes that occur in brain tissue. And so with that, it follows that organelle segmentation and analysis will also be a valuable part of scale-up in 3D EM connectomics as we go forward. With that, I'd like to thank the people at the Sung Lab at Princeton. With that, I'd like to thank all of the people at the Sung Lab at Princeton University, as well as those in the Allen Institute for Brain Science and Baylor College of Medicine who worked really hard to put this data set together. And I'd also like to acknowledge our funding sources at IARPA and the National Institutes of Mental Health. Thank you for your attention. And now I'd like to introduce the next speaker, who is Luis Rodriguez.
Hello, everyone. My name is Luis Rodriguez, and I'm a member of the Johns Hopkins University Applied Physics Lab. Here, I'll briefly discuss some of the questions and answers that as a community we will have to consider when addressing storage challenges and ensuring data democratization at the petabyte and exabyte scale. From our understanding of the 302 neurons in the C. elegans, which amount to about 1 terabyte of data, to the billions that make up the human brain amounting to roughly 1.3 zettabytes, our community will face increasingly larger challenges. We're aware of a number of fundamental challenges now, but there are unknowns that will only emerge as we move forward larger data scales. This includes collecting, uploading, and sharing data. So let's start there. As researchers in the community produce data of different modalities and organisms, we need to think about where to store it to facilitate the sharing and access of the data and results. As the rate of data generation increases, more bottlenecks will emerge. Take, for example, a Carl Zeiss multi-SEM microscope, which can produce about 1.5 terabytes per hour. At that incredible rate, we would still need more than a month to finish imaging an entire brain. So even faster processes are still needed, and our storage infrastructure needs to be able to keep up with those speeds. Given the challenges associated with standing up and maintaining the required physical storage, cloud and serverless approaches provide an attractive option. So fortunately, there are a number of current data approaches in the community that support different workflows, tools, and scientific pursuits, each of them having their various strengths, like Janelius Divid, which allows for versioning, BossDB, which provides authentication and serverless computing, and Cloud Volume, which has a built-in meshing infrastructure. I'm actually lucky to be a member of the APL Brain team building BossDB as a part of the Brain Initiative NIH Data Archives program. So the inspiration behind the Block Object Storage Service database, or BossDB, is to provide a platform for users to upload and store volumetric data at scale. The BossDB offers a number of services to enable scientific workflows and discovery. So scientists can upload data using our ingest client, which leverages a plugin infrastructure to broaden the scope of data formats and support ingest of data sets over a petabyte in size. And we also have developed Intern, a Python SDK that provides data access to support scientific workflows. Intern allows us to create and get data cutouts from the Boss using the BossDB API. Similarly, it allows us to access data from Divid and Cloud Volume. This synergy between data infrastructures allows us to have the best of all worlds, making it seamless for us to share data across platforms and allowing us to use unique data services. For example, we could grab an object cutout from the BossDB, update and version it using Divid's versioning API, and then generate a mesh using Cloud Volume, all while caching the data locally with an embossverse instance. Like with other platforms, to visualize BossDB's volumetric raw and annotation and mesh data, we can use open source tools like Neuromancer. And to browse publicly available datasets on the Boss, we maintain a website, bossdb.org. And as we develop BossDB and handle petabyte data at unprecedented data ingest rates, we noticed there's still some unanswered questions. Like, for example, as a community, these are some of the challenges that we should be aware of as we move from a terabyte to a petabyte to a zettabyte scale. Should we be meshing on demand such that only meshes of those segmentation IDs that are requested by the user be presented? Or perhaps this could potentially reduce the cost of storing the actual meshes while retaining the advantages of the annotations. Should we take a step further and segment on demand? Segmenting only areas of interest and building from there may be more cost efficient. Should we maybe also just mesh in a volume-based way where we segment the entire volumes, or do we gain more from segmenting a single process in the entire span of the data set, which might be more efficient? As we store all this data, we also need to think about the resolutions. Res0 data is rarely used to run segmentations algorithm, so is it interesting if we just downsample it and then not keep it? What's the lowest resolution that is actually useful to researchers? And how do we make the ingest process easier? Because as we move towards larger data sets, our infrastructure needs to be able to handle many more writes per second in order to ingest the data as quickly as possible. Finally, how do we make sure that as our data size scales up, we continue to make groundbreaking discoveries? We hope to continue this discussion with you in our BossDB Slack, to which you can find the link on this slide, our last slide. As a team, we would like to just thank IARPA Micron's program, along with the NIH, for funding the BossDB and Saber projects, and I'd like to personally thank the APL Brain team with whom none of this work would be possible. And finally, a big thank you to the organizers of this symposium. Thank you very much. I'd like to just introduce our next speaker, Denise Cazares.
Hello, my name is Denise Cazares, and I am a rising junior at Johns Hopkins University. I am majoring in cognitive science with focal areas in computational approaches and neuroscience. Today, I am representing one of the circuit program cohorts at JHU's Applied Physics Laboratory as part of APL Brain. I'm going to share with you some information on our mission to democratize the field of connectomics. Before I delve into our project, I want to briefly acknowledge and introduce CIRCUIT, which stands for Cohort-Based Integrated Research Community for Undergraduate Innovation and Trailblazing. It is a program for trailblazing students with high potential but that are having difficulty achieving their goals because of various barriers in their way. The goal of the CIRCUIT experience is to educate future leaders in areas of STEM, such as data science, machine learning, and artificial intelligence. There is a holistic approach in recruitment, and it is a 14-month commitment with training in technical areas, professional development, and team-based projects to make significant contributions in the world of science. There are challenges that are important to consider in the scope of this mission. The first is scalability. With advances in technology, big data is ever more pervasive and necessary for the kinds of questions being investigated. Big data, in this case, is at the petascale level, and to be able to handle this amount of data, we are having to rely on cloud computing resources. Another aspect of scalability is to be able to customize workflows into more digestible chunks. The second challenge to consider is reproducibility. It is necessary to be able to reproduce results from colleagues to be able to further the field. This helps generate baseline connectomes for comparison in other experimental conditions. Accessibility is the third challenge. Computational complexities often get in the way of running workflows on these large datasets. Because of this, an intuitive tool is desirable for connectome generation and analysis. In addition, access shouldn't be limited, so an open source approach would help to democratize connectomics. To reiterate, there is a high demand in the field to streamline the process of generating connectomes and graphs for analysis, but in a more intuitive manner. APL confronted the challenge of scalability by developing a state-of-the-art toolkit and volumetric database, Saber and BossDB. For scalability and accessibility, we propose a web portal that merges the capabilities of both ecosystems into an accessible centralized location. This streamlines the process for reproducible assessment of existing and novel electron microscopy neuroimaging datasets. This scientific portal will allow researchers to focus on answering the questions that will inform our understanding of the connectome. The user-friendly graphical user interface caters to researchers with diverse computational backgrounds. Writing code is no longer required to run a workflow, since user specifications in the portal can easily be translated into scripts. Our approach can be simplified in the following schematic. We created a web portal that can authenticate with BossDB to access what are currently publicly available datasets. Next, users can initiate a new job. Current capabilities are with the public datasets, but we are developing capabilities for users to upload their own datasets for processing. Then users specify what processing tools they wish to use on the dataset. The workflow specified will determine which parameters can be customized. After parameters are filled in, the workflow is triggered and the specific interdependencies and order of tasks are handled in the background so the user doesn't have to deal with them and to simplify the process. As the dataset is processed, there is a periodic check-in to see if the workflow is complete with API calls to the Apache Airflow task scheduler. These are large datasets, so the processing can take some time, from a few minutes to a few hours, depending on the complexity. But with the ability to customize the workflows, it is easier to minimize wait time and still extract valuable information. Once the workflow is complete, it is returned to the user to download or visualize in NeuroGlancer. There are some preliminary analysis tools in development that are being made more user-friendly. I want to thank the BossDB and Saber teams for facilitating collaboration with us and for their ongoing support through the project. Lastly, a big thank you to all those involved in today's presentation. Next, I would like to introduce Jennifer Stitzel.
Hi everyone, I'm a fourth-year PhD candidate at the University of Pennsylvania. I'm in Danny Bassett and Tim Lucas's labs, and today I'm going to talk about using epileptic spikes to understand how human functional brain networks respond to perturbations. So I want to start by talking about why I chose an epilepsy project for the symposium. And it's because I really think that epilepsy is well characterized as a network disease. And I think that's largely because a lot of the features of epilepsy have to do with synchrony between brain regions. So the only consistent and defining feature of epilepsy is a predisposition to seizures. So we have an image of a specific kind of seizure, a generalized seizure up here on the right. Seizures will generally start at a specific brain region called the seizure onset zone. It's indicated by this black circle here, but it will very quickly spread across the entire brain. So you can see these really synchronous, high amplitude oscillations across the entire brain, where each of these lines here is a recording from a different brain region. So that's a seizure. There are some other interesting features of synchrony that are prominent in epilepsy as well. So for example, different phases of seizures have different patterns of synchrony, and you can even see these changes hours before seizure starts, and they can be predictive of when a patient is going to seize. So because of that, I do think epilepsy is well suited for tools from network neuroscience, network science, and complex systems. And essentially, to use these tools, what we're going to do is characterize the brain as this set of discrete brain regions indicated on this picture by these grayscale circles, and the connections between them, where the connections between them here are this, they're going to be like this synchrony that I've been talking about. So essentially, it's just how similar the activity is between brain regions. I think that this framework can be useful for translational research, but also just for basic neuroscience research into characterizing the brain as a complex system. And my talk is going to focus on that latter part. And in complex systems research, once you've defined your system, a good way to study it and learn about its properties is to observe how it responds to perturbations. So if you change one of these regions, the activity, how does that affect the rest of the network? And epilepsy gives us a really unique opportunity to study this because of a specific waveform that we see in epilepsy called an epileptic spike. So now we see a spike on the right. It looks different from the seizure. It still starts or tends to start in the same region, so it's also starting at the seizure onset zone, but you'll notice that the spike is localized. It doesn't spread to the entire brain. And while most patients don't notice when these spikes are happening, there is some evidence that they're having some kind of distributed effects. So for example, some other features of brain activity, such as spindles, will be affected by spikes even when the spindles are located outside the seizure onset zone. Additionally, spikes will affect task performance if they're happening while the patient is engaged in something cognitively demanding. So despite this evidence that these spikes, even though they're a focal artifact, are affecting the entire brain, the extent to which they perturb these sort of global measures of synchrony has not yet been quantified. And I think there are a lot of reasons for this, but one major challenge standing in the way is that epilepsy is very heterogeneous, and I think answering this question really thoroughly would require, first of all, high quality electrophysiological data where we can estimate the synchrony between brain regions well, and also a lot of people so we can get something close to a representative sample. So luckily for us, this data and all of those tools are now available, and so we can start to answer this. So we're going to be using data collected by a really big collaborative group that was led by Mike Kahana at the University of Pennsylvania, of 250 people with electrocorticography data. Electrocorticography is a source level recording. It's electrophysiological data recorded from electrodes that have been implanted into somebody's brain. And for this project, I'm going to show you some preliminary data from about 180 of these 250 people. So specifically, what we're going to do is we're going to take all this data, we're going to pre-process it, and then we're going to try and automatically find where these epileptic spikes are. So you can see they're indicated on this toy time series with these gold circles. After that, we're going to break the data up into 1-second windows, and we're going to break the data up into frequency bands. This is a common thing to do with electrophysiological data, where we break it into slow all the way to fast oscillations. And then within each frequency band and within each window, we're going to look at a bunch of different estimates of functional connectivity. I'm only going to talk about 3 of them here. The first one is an amplitude envelope correlation. This is essentially just a correlation. It's telling you how similar the amplitude of two brain regions is over time. The next one is the phase locking value, which has nothing to do with amplitude. All the phase locking value is telling you is whether two regions have a consistent phase offset over time. And then the last measure is coherence, which is sort of like a mix between the two. It's quantifying similar phase, but it's also weighted by the amplitude of each brain region. So once we have all of those functional connectivity measures, what we're going to do is use robust regression to try and estimate the effect of these epileptic spikes on the strength of the functional connectivity. And we're going to have two different predictors to estimate this. One of them is binary and just says in each time window, is there a spike or not? And the other one is continuous and gives the number of spikes in each time window. And the last thing I want to mention before getting into the results is that we are also including power as a covariate. The band-limited power is just a way of estimating the amount of brain activity. So any changes that we see are going to be above and beyond what would be explained by just changes in brain activity resulting from the spikes. Okay, so this is the format that I'm going to present most of my results in. It's a lot of data, so I want to kind of go over the axes of this first. We're going to have our two different predictors, the number of spikes and the binary, just presence of spike on the y-axis, and then all the different bands are going to be on the x-axis, and each measure is going to be a different color within these plots. Okay, so first thing we want to do is just address this very basic question of are there effects? Do epileptic spikes impact functional connections in the brain? And what we see is that these red asterisks are indicating significance. They're not localized to specific bands, but instead are localized to specific measures. So what we see here is that selectively in these amplitude-based measures, so the amplitude envelope correlation and coherence, we see that a single spike will disrupt these functional connections and leading to decreases in the strength. And then more spikes after that will sort of build that back up again. And I do want to mention that all of the bright red asterisks replicated with another spike detector, as well as an orthogonalized version of the amplitude envelope correlation. Okay, so the next question we want to ask is whether these effects are really driven only by the seizure onset zone where the spikes are starting, or if they're spreading to the rest of the network. And so to do this, rather than just looking at raw effect sizes, what we're going to do is get the value from the seizure onset zone and subtract it from the value we get in the rest of the brain. So this plot is now going to show a difference between effects rather than just the effect itself. And what we see is that for most of these, we get a really tight distribution around 0, so there are not very big differences. None of these are significant. After multiple comparisons correction, these gray asterisks are indicating things that were significant before multiple comparisons correction. So the interpretation here is that it seems like these effects really are affecting distal regions, even though the spikes themselves are extremely focal. Okay, next, what we want to do is try and parse some of this variability across all of these individuals. And so one potential source of variability could be where these spikes are originating from. So if you look at these toy brain networks, you might imagine that a spike that starts at this orange node would affect the network differently than a spike that starts at this green node. And a lot of the theory in complex systems says that's because these two nodes have different connections, so they're going to affect the network differently. So we wanted to try to test this in a pretty basic way that was going to be robust to electrode localization, because that could be a noisy process. So what we're going to do is compare the seizure onset zones by hemisphere. So language lateralization induces a lot of changes in connectivity. So we're going to test if there's a difference in these effect sizes for subjects that have their seizure onset zone ipsilateral to their dominant hand as opposed to contralateral. What we see is that selectively in these lower frequencies, the theta and alpha bands, when the seizure onset zone is ipsilateral to their dominant hand, we get larger effects. And so one potential interpretation of this is that something about that contralateral hemisphere, something about the architecture of it is preventing the further spread of these focal perturbations. And in the future, I'd really like to dig into this a little bit more and get a finer scale breakdown than just the hemisphere. Okay, so the last thing that I'm going to talk about today is whether the magnitude of these effects on resting data predicts task performance. So in this data set, individuals completed, some subset of 5 tasks. And since performance on all those tasks is really highly correlated and they're pretty similar, what we're going to do is just take the individuals who completed a couple of them and get the average of their performance. And what we find is that selectively with beta coherence, we get a significant effect where a larger change to functional connectivity resulting from epileptic spikes actually leads to better performance. And so in order to get a more meaningful interpretation out of this, I'd like to analyze some past data and potentially do some statistical causal modeling and also look for mediation by some different clinical variables. So that is in the works. So as a really quick summary, we've characterized the brain as this functionally interacting complex network. We've shown that these focal perturbations spread and affect the entire network, that effect is mediated by where the spike starts, and that in some cases, the magnitude of that effect tracks with behavior. And now I'd like to thank everyone in my lab, everyone at the hospital, and the people who collected the data for helping me complete this project. And now I'd like to hand it off to the next speaker, Dr. Kristen Harris.
Good morning. Thank you very much for inviting me to present some of our work at this meeting today. I'm going to be talking to you today about enhanced resolution for 3D electron microscopy analysis of synapses. And this work is powered by our NSF NeuroNext Award, and I'm very grateful to the NSF for this opportunity. So my goal today is to fill the resolution field size gap with scanning electron microscopy operating in the transmission mode, which we are expanding to tomography on the scanning electron microscope. So scanning electron microscopy in the transmission mode, or TSEM, has been really developed on standard instrumentation, in this case the Zeiss Super 40, and was published in our paper back in 2013. Transmission scanning electron microscopy allows us to have very large fields, much larger than the standard transmission EM field on the upright microscope. As you can see, we just needed to have about 6 or 7 fields to montage the entire dendritic arbor of a pyramidal cell, for example. And yet, while operating in the transmission mode, we maintain the 2 nanometer XY resolution that's needed to visualize small structures like polyribosomes, microtubules, vesicles in detail. Well, what we want to do is to be able to integrate this cell biology of the neuron and synapses into understanding neural circuits. So the first thing that we're doing is improving alignments of these large fields using the SWIFT-IR in a GUI that's been implemented in our laboratory in collaboration with our colleagues out at the Salk Institute and the Texas Advanced Computing Center. So if you look at the quick video here, the original absence of alignments on the left, the field that's going to be working with is in the middle, and the aligned version is over here at the right. And so the user gets to enter all sorts of features to improve the alignment, check the alignment, do it at different scales to make sure that it's absolutely perfect by the end of the day. So we need also, though, to have EM tomography to reveal substructure that's buried in the sections that are cut to visualize in an electron microscope. These are 2 serial sections from the same dendritic spine that has a spine apparatus, that critical structure I showed you that seems to organize the principles of long-term potentiation of synapses, in 2 adjacent sections. We really want to know how much surface area is ascribed to this lipid-producing, calcium-regulating organelle. We would really like to be able to see those structures in detail and be able to not have this overlap of these gray walls that are shown here on adjacent sections. We also need EM tomography to overcome the fragility of the thin sections that are photographed either in the transmission electron microscope or typically in the scanning microscope using backscatter. And these sections include, these artifacts include folds, which you can see here, or tears that occur in these fragile sections. And this is one of the biggest problems with alignment and autosegmentation that is going forward. So, but the tomography that is available right now is that's in the upright transmission electron microscope, and it's really quite limited by the small field size to about 1 micron squared. But you can see in this very thick section, this is a 150 nanometer thick section, that there are structures that can't be resolved until we make the virtual section. So I'm just going to show you that same field where virtual sections have been calculated from a tomogram, and you can see in quite detail the membranes of each part of that spine apparatus. You can see the vesicles very clearly. You can see a microtubule. You can see where the extracellular space is. And you can even see a clear example of a polyribosome emerging on these sections here. So TomoSem is really using tomography now on these large scanning electron microscope fields operating in the transmission mode. These fields, a single field can be up to 10,000 microns squared, 100 microns on a side. We can have the much thicker sections because we can do tomography. We've tested up to 300 nanometers now. So for the same volumes that we typically do 100 serial sections, we only need to collect 40. That's less human cutting time. They're stronger sections. They do not require any post-section staining, so we have the homogeneity of en bloc staining. And there are many fewer or almost no flaws. We've yet to see any flaws in these thicker sections of the nature of folds and tears. Then we calculate virtual Z sections, and that will reveal the buried structures and provide for better autosegmentation. We estimate right now that we need about 25 images per section through the tomogram, and over 40 sections, that's about 1,000 images taken. It's automatable for multiple image, multi-section, and multi-grid. And this is an example of a 250 nanometer section. We're developing this on an existing platform, the Zeiss Merlin Gemini 2. It's a relatively inexpensive instrumentation that is affordable at the level of a startup package, has the A-stem detector, custom grid holder, 5-axis eccentric stage. Here's this 250 nanometer thick section in the transmission mode. You can even see your regions of interest, and that 15 kV provides good contrast throughout the section depth. The TomoSem operates now in a conical tilt tomography. We have a single tilt, one dynamic focus transform, conventional stage and detector. The rotation is up to 220 degrees around a single detector. We're now trying to get rotation for 360 degrees with a second detector. So here's a short movie that illustrates this this custom grid holder, which is mounted on a 5-axis eccentric stage. The A-stem detector is below. The Merlin field emission SEM field emitter is located above. And what happens is we simply make the rotation, the tilt, and then one gets the cone by rotating about that single tilt. We go to the region of interest and look up close. You watch, for example, as we rotate around this particular synapse. You can see there are planes in which it comes into perfect cross-section focus. And we are now in the process of developing, our next goal is to compute the virtual Z sections at a resolution of 2 nanometers in XY. Z, 10 to 15 nanometers will disambiguate all structures that are overlying and any of the buried structures. And these will be our XY fields for a total SEM volume of about a cubic millimeter. So our goals have been filling this resolution field size gap with TSEM and TomoSem. And I encourage you to see the virtual poster by James Carson at this Brain Initiative meeting. Thanks to everyone who's been involved. And next up will be Eva Dyer. Thank you.
Hello, my name is Eva Dyer, and I'm an assistant professor at Georgia Tech and Emory, and today I'll tell you about the work that we've been doing in my lab to use deep learning and machine learning approaches to build multi-scale maps of brain structure and organization. All right, so to motivate a lot of the work that we do in my lab, as well as, one of the key motivations of this workshop is being able to handle the sort of scale and resolution that we're currently dealing with in many of these new imaging modalities, including electron microscopy and all different types of microscopy data sets. And so with these large data sets, we're often faced with an issue of trying to figure out and interpret what's going on within these data sets. And most of the time, we're actually facing with raw images that we need to translate into scientific insights. So the key kind of driving question behind the work that I'll describe today is a question that we are often faced with when dealing with many different types of neuroanatomical studies. And in particular, our question is, what makes brain areas distinct? When looking at an image of many different brain areas, there are, key features in the underlying microarchitecture or anatomy that, strike us or stand out. And based upon those observations, this is actually how we go about as anatomists defining what constitutes different brain areas. So in this example, I'm just showing a large-scale X-ray microtomography data set that was captured at 1 micron isotropic resolution. And here I'm showing a sample that spans all the way from somatosensory cortex down to hypothalamus. So it's a very diverse sample. And if we zoom into all the different regions therein, we see that distributions in axons, which are shown here in bright white, distributions of cell bodies or blood vessels are strikingly different. And so these are really the key questions. And so we were interested in whether or not we could actually train a neural network or an automated solution to be able to find these patterns without having to take in training data that tells it necessarily what to look for. So to do this, we developed a tool which we call Deep Brain Disco or Deep Brain Discovery. Essentially, the idea is shown here in this overview slide. So what we do is we start by zooming into very small local snapshots within these different brain regions. We essentially train a neural network to then discriminate between these 6 different brain regions shown over here on the left. So discriminating between regions of cortex or striatum or thalamus or different paths of white matter. After we train this neural network, we then go and we actually open it up. So we open up this black box and we actually use the neural network as a feature extractor. So instead of using it just to classify the brain regions into different areas, we're extracting the features from this network. And then what we do is we use applied mathematics tools or matrix factorization techniques to then take those representations from the network, rotate them, and embed them into a lower dimensional space. Once we have that, we can actually start to zoom in to regions of the sample that we might not have already trained the network to discover. So this is now just showing some kind of highlights of the results from this strategy. So what I'm showing here in the middle are overlays now where we're embedding these network activations onto different parts of the brain. And so what we see immediately is that our network, it has been trained to discriminate across these different areas. It can do a good job initially of just dividing the brain regions into parts. But we also see some really interesting effects like different parts of cortex being linked with different parts of thalamus, which is something that, the network hasn't been trained to do. We can zoom in even further, and if we actually look into these representations within cortex, we find that there are laminar differences, or basically the layers within cortex have been revealed through this particular strategy. And we can zoom in even further and find that this network has actually pulled out some interesting parts of the data that correspond to higher or lower cellular density regions. So essentially what this network has done without being told to do so is it's found a lot of interesting patterns of microarchitecture directly from the data using these data-driven representations. We can also expand upon this and actually train the network to also segment at the pixel level, which is something we've seen in some of the other presentations today. And so we can create now a multi-scale map of both different brain areas and the underlying microarchitecture therein. I just wanted to quickly say that we've released this data through the BOSDB, so bosdb.org. This is an open access, publicly available X-ray microtomography data set with tons of heterogeneous brain areas, and we have multiple annotations and segmentations provided as well. And in summary, we've provided a new neural network solution for being able to extract features from brain images and reveal things like different cell densities or even barrels within cortex. We can couple this with other structural segmentation tools to find a multi-scale map. And we've also told you a bit about this new interactive X-ray micro CT atlas and segmentations that are now on BossDB. And with that, I will acknowledge our team and also our collaborators, including Will Gray-Roncal at the APL, who also hosted the symposium. Really great opportunity. Our funding sources, including the NIH. And I'd like to thank you all for your attention. Thanks. Up next, we have Steve Plaza.
Hello, I'm honored to present on behalf of the Genelia Fly-EM research team at Genelia in collaboration with Google. Our group has been working on connectomics for about 10 years, obviously in the Fly, as our name indicates. And our group has really worked on the whole pipeline, the whole infrastructure from imaging, acquisition of the data, to alignment, segmentation, synapse annotations, and producing the connectomic product. So it's almost like a factory, and we've produced, I think, a lot of connectomes over the course of the last several years. Prominently, in the last few years, we've worked on the sample called the hemibrain. This contains a large portion of the fly brain, the fly central region. And the goals of us analyzing, of reconstructing a connectome for this volume was to understand various behaviors and properties of the fly, such as associative learning, sleep, understand navigation better. But there's another goal, which is to facilitate science in the community and to make the data set accessible. Now, one thing I just want to point out quickly is that this is a dense connectome, so we're trying to get all the neurons, all the connections as much as we can for a given region. And as you could see, in this movie, you don't look at this movie to understand or to see the exact details. There's a lot of stuff going on here. We're only showing a subset of the neurons, but I think the thing to highlight here is the complexity. And this data set, even though it's just a fly, in the spirit of this PETA and Exascale volumes that we're looking at, has over 20,000 neurons that we've reconstructed. There's tens of millions of synapses in the data set. We're only showing a small section of the data here because it's very overwhelming to look at, frankly. And I think that's an important thing to remember when we're talking about these connectomes is that I think there's a lot of great technology that's been developed, but there's actually not that many connectomes. We heard about, obviously, Sidney Brenner and the worm scale. There's a couple other small model organisms. And then the fly now is the biggest one that's out there. The challenges that are involved is actually not so much in even acquiring the data and segmenting data, which, of course, is a big challenge, but it's actually generating the connectivity graph itself. It involves a lot of manual labor, a lot of proofreading in the process. And with better segmentation, of course, we expect the validation time to be significantly better. But still, for the N of 1, for the first time that you do something, it's going to require validation time. And so we've heard from Jeff talk about a lot of the challenges that are involved, but I do want to emphasize that the grayscale, the EM data, the segmentation even is a means to an end. The end is what you can analyze in the connectome. And I think actually that is the biggest challenge, is to make it the most consumable possible. You can see here that we developed a few tools to help with this. One of the things I just want to highlight is some of our proofreading tools that we worked on. These are high-level tools for looking at neuron shapes and trying to figure out if there's any errors in the reconstruction. Now, we show a little inset here of grayscale, but notably, it's not even clear that we really need grayscale many times to do our proofreading. And actually, one of the longer-term goals that our group has is to try to eliminate even needing EM data to reconstruct the connectome. So you could imagine the connectome and the neuron shapes could potentially even fit in a laptop, even if it's a mouse scale connectome. And so that type of model order reduction, I think, is critical for making the data sets accessible to the community. I won't talk about synapse validation, but we do have a rigorous system of iterating with machine learning and spot checking those results. To make the data accessible, we try to split up the volume into several canonical brain regions. We can use the connectome to help guide this process. It's a little bit of breaking things down hierarchically. We define cell types to also break down the complexity of the data. Now, cell type's a little bit of a slippery term, but there are different ways that you could use connectivity to help at least tease out some differences between neurons that might otherwise look morphologically very similar. And then also we've introduced a tool called Newprint to help us analyze this data. This is a graph database that we can use to represent the data so users can navigate very quickly. But importantly, we also store the data in a hierarchical way, so we can look at regions of interest and bore down into the connectivity graph or more detailed skeleton data if needed, which, as it turns out, most of the time people don't really want. As beautiful as EM data is, most of our users just care about the result. And I think that's some of the lessons that we've learned is that it's very important, I think, to have a very agile mindset. We've done a lot of rapid prototyping. We did a lot of iterations. We try a lot of volumes at small scale and to make sure that it scales up. But also the management's very critical. The mission before the papers. We always focused on getting the connectomic result. We have a mixture of labs that work with us with some kind of like centralized guided steering committee. And I think those types of infrastructure get more and more important as you deal with these diverse interdisciplinary teams. I would like to thank you for your attention and now introduce Nunu de Costa for the next talk.
Thank you for the opportunity to present in this symposium. My name is Nunu de Costa, and in my presentation, I'm going to discuss linking cell types with electron microscopy-based connectomics. And for that, I'm going to use a set of data sets collected under the IARPA Microns program in a collaboration that has been co-led by the Allen Institute, Princeton University, and Baylor College of Medicine. So cell types are one of the most fundamental concepts in neuroscience, and connectivity connectomics can contribute to it using connectivity, of course, but also the single-cell morphology. And you can see here in the center an example of a beautifully segmented neuron from one of our EM data sets done by Sebastian Zwing's lab. However, there are many other properties that are not immediately registered with connectomic data sets, as for example, synapse physiology or transcriptomics. However, and increasingly, together with these other data sets, morphology is also collected. And so morphology can be used as a Rosetta Stone between these modalities and connectomics. This is by far not the only way to link these modalities, but it is a way that is possible right now. And as a first example, I'm going to use our recent study on the structural and functional logic of Sandlia cells. And in this video, you see the reconstruction from EM. In gray is the axon initial segment of layer 2 pyramidal cells in the visual cortex of the mouse. And in color, there are many different Sandlia axons. And you see that there are some pyramidal cells that receive very few synapses, while others receive even up to 10 times more. And the connectomics part of our study allows us to map the complete connectivity between these two cell types. But actually, one of our major conclusions that the Sandlia cells carry a global inhibitory signal that is adjusted to each one of its targets will not have been possible by connectomics alone. And so using morphology, we're able to link our connectomics study with the transgenic cell lines, with the transgenic mouse lines. Using these mouse lines, we were able to perform some of the first awake behaving recordings of Sandlia cells. Now, for other examples, I'm going to use a different data set. This is a much larger data set. It's on the petascale range. It's a cubic millimeter of cortex collected with transmission electron microscopy. It goes across several visual areas. And you see video an example of some of its reconstructions. It contains approximately 100,000 neurons with soma in the volume. And here you see a few examples of a layer 2-3 cell in RL in dark blue on the left side, and some of its targets both focally and distally. And now what we can do, and what Forrest Coleman did, is to classify neurons in this data set using basically the same methods that were used in studies with from slice physiology. And in this case, you can see here results of a study of Goins and Sorensen et al. From 2019. So in color, you see the morphological clusters of cell types from the slice study, and in black, you see the results of the morphological classification from the REM data set, and you can see that they match pretty nicely. And that's what that allows us to do now, is from the connectomic side to map the connectivity patterns of this neuron. And here's an example collected by Agnes Plodder across several layer 5 ducted cells. And from the slice work, we can connect synaptic physiology, electrophysiological properties, or transcriptomics with patch-seq. And morphology allows us to link these modalities. And we think that will be a very powerful link. So to finalize, I want to say that some of our data sets are already publicly accessible to everyone. Please use them. The more you use them, the more you encourage us and others to release the data to the public. And also to say that even though I talk with neurons, EM data sets are much richer than neurons. There's glia, there's vasculature, there's organelles, as you've heard earlier from Alicia's talk. And to finalize, I'm just going to show a short video, again, from a beautiful automatic simulation from Sebastian's lab in this data set. In purple, there's a microglia next to a layer 2-3 pyramidal cell. And to end, I want to thank everyone, particularly Clay Reed, which is the head of our department at the Allen Institute, but also our collaborators both internally and externally, particularly the lab of Licensing and Andreas Elias, and of course our founder, Mr. Paul Allen.
Great, thank you all to all of our presenters who did a great job of presenting on the current state of art for the field, both for science and engineering, as well as framing some of the challenges that we foresee as we move forward and as the scale for these data sets increases. My name is Brock Wester. I'm moderating this session with Will Gray-Roncal. We're now in the panelist discussion, question answer session of the meeting. We got some questions from the audience during the presentations, but as a reminder, please continue to submit questions for panelists and we'll answer as many as we can within the session. And we'll start with some specific questions to each of the panelists. Jeff, you talked about the benefits of building a connectome for an entire mouse brain. How would you compare the importance of this goal to that of comparative connectomics, where we image smaller regions from multiple organisms in different scientific studies?
Thanks, Brock. I think both of those goals are admirable, and I couldn't qualitatively say one is better than the other, but it's important to realize, I think, that doing a whole animal's connectome offers a kind of information that is never available by doing small regions. And that comes from the painful realization that no matter how big a region of the brain you look at, a substantial number of the neurons that are entering that region are coming from cells that are far-flung, sometimes many, many millimeters away from the cells you're looking at. And for that reason, if one ever wants to have a sense of how neurons that project from one part of the brain to another make synaptic connections on which particular cells and which particular layers, for example, one is going to have a whole brain at one point or another. That's not to say there are not very important questions with comparative connectomics, and maybe in a subsequent question I'll talk about that.
Great, thank you, Jeff.
I was just going to say, maybe as a follow-up to that, Nuno, I wonder if you might have thoughts on which approach makes the most sense.
I was just going to say that I agree with Jeff, but I would like to add that in the process of scaling the technologies to be able to collect exascale mouse connectome, there's going to be many petascale data sets that are going to be needed in that process, and we'll basically, I think, will make generating petascale data sets a kind of standard, therefore allowing also those comparisons across inter-individuals, inter-species, inter-areas, and so forth. So I don't see it really as a one or the other. I see it as them almost happening simultaneous and in the process of scaling up to the exascale mouse connectome.
Thanks so much. Okay, Eva, what is the role of other imaging modalities other than EM to aid in scaling up the reconstructed connectomes or capturing neuroanatomy from larger organisms?
Hi, yeah, it's a great question. I mean, I think today we heard a lot of talks that focused on electron microscopy, and then I also spoke about X-ray microtomography, which is essentially using a similar preparation to EM to achieve a dense labeling of essentially, well, in the case of EM, as we saw in many of the talks, I mean, every single piece of membrane, bound tissue, so you can see everything, and it's a very dense reconstruction. But, in terms of a lot of other emerging technologies that use sparse labeling techniques along with light microscopy, we're able to augment this dense view, right, that we get from EM with other types of information or antibodies, information about cell types. And while we might not see that in the same dense picture, we can imagine a lot of tools being developed in the future to integrate this sort of dense and also sparse understanding of the underlying microarchitecture and anatomy. And so, yeah, I think moving forward, I imagine that we'll see a lot of new emerging tools that use both experimental and also computational techniques to build multimodal and sort of multi-dimensional views of the brain and also the connections and circuits therein.
Thanks, Eva. So maybe our next question for Kristen Harris. So Kristen, of course, collecting all this big data is necessary, but not sufficient. When we think about that process of acquiring the raw data and processing it and doing analysis, what should we be thinking about as a community to ensure that we're collecting the right data and helping to make it available to the community for scientists and engineers?
Thanks a lot for that question. And as many of the people in this group will know, that I have been really pushing hard to collect this data at 2 nanometer XY resolution so that the kinds of data that we saw Alyssa present, but even more refined data, can be studied in the context of cell biology in the future. And so I really am pushing hard that we don't go after a lower resolution data set that will maybe get us the synaptome, but not get us the synaptic weightome, and not allow us to really understand how the cell biology of those connections works. So that's one of the things that I'm pushing really hard for, and I know that that's going to increase the amount of time it takes to get this, but the other beauty of pushing for that resolution is that I think in the same vein that Nuno brought up, it will provide the development of much better tools along the way for analyzing the data, and furthermore for segmenting and training tools to segment it. The better the resolution of the images, both in X and Y and ultimately in Z, will make that aspect of it much more reliable and faster. And so I think we are soon going to come to the bottleneck at the analysis end, not at the image acquisition end. And so I'm hoping that what all those who begin to work on the mouse connectome will do is consider the importance of having really high resolution data as they collect the data. The other point that I make over and over again is that collecting a lot of metadata before the mouse that gets selected, or the 10 mice that get selected for creating the mouse connectome, collecting a lot of metadata about their behavior, their development, the normality of their brains relative to other animals at other scales, will be really critical, I think, for people to be able to view it as an atlas that new data coming up can be scaled against that metadata from new animals, and not simply rely on the anatomy as the component of the atlas that will ultimately make this data really outstanding. Thank you.
Great, thanks, Kristen. This next question will go to Steve. During the symposium today on neuro AI and neural dynamics, the discussion talked a little bit about the potential metric for assessing data quality or usefulness of a neural data set to current or potential scientific questions or challenges. How can we go about rating these imaging and connectomics data sets to benefit the community?
Thanks, Brock, for the question. It's definitely a challenging question, because I think that the biggest thing is sort of what is important for people, and sort of what I've been finding and working with EM for several years now is that I often find that people don't have the right questions in the first place. There's a lot in the data set. There's a lot that you can view in the data set that is much more nuanced, much more intricate than what people are asking. So I think there are definitely some advanced things that we could do. But for at least questions that people ask, what we've done practically before, let's say if you care about whether you can see all the synapses or mitochondria, whether you can do the connectome accurately, we try to practically assess the situation by measurement. So obviously this would entail maybe imaging something in different modalities, or trying things at different resolutions and seeing if we're missing anything at our production scales. For the connectome that we had done in the fly, we started out with small regions and we scaled up. And each time we scaled up, we measured very carefully what we saw before to make sure that we could extrapolate those techniques with bigger data sets of so-called similar quality. But like I said, I think the question is the question, and trying to understand exactly what we want is critical.
Great, thanks, Steve. And so I think next up we have Nuno, who's responding to an audience question from Terry Sijanowski. And so Terry asks, the cost of sequencing a human genome has decreased exponentially from $1 billion to $1,000 in 20 years. And he wonders if connectomics will benefit from a similar exponential decrease. So this to Nuno first.
Yeah, I think that is very much possible. Currently, a large proportion of the cost on the data is not so much the acquisition, but is the storage of the data and all the computation required to segment the neurons and their synapses and their organelles from the data. And so as that cost of computation decreases, so will the cost associated with collecting this data will decrease as well.
Thanks for that, Nuno. And Steve, I wonder if you might have some feedback on that question from your experience at Janelia.
And from my perspective, the dominant cost has always been the proofreading. So again, I think the reason oftentimes the other costs are more visible is because that's sort of still where we're at. It's very hard to extract the connectome. I think there's a few things that we can do to lower the costs across the board. At least for the imaging part, we've benefited technologically speaking when industry is involved. So if there's some way to convince computer manufacturers that building microscopes is a great thing, somehow gets into their bottom line, that would be one way to really reduce costs on some of the acquisition. For machine learning, yes, compute will go faster, but I think one of the things that we could do is that a lot of times we're wasting a lot of compute cycles for things that are easier. So maybe doing our compute a little bit smarter to make sure that we're targeting the compute that we really need for areas that are a little bit harder to analyze is a good way to reduce costs there. And then finally, for proofreading, I think we're just seeing that as a consequence of the improvements upfield. In the last 10 years, we started out where a fly connectome would require maybe even tens of thousands of proofreader or editor years. and we believe we have that down to maybe a couple hundred years. So we're looking at, you know, a couple orders of magnitude just in the last 2 years, or 10 years. And I think we could probably get another few X as we do our next connectome in the next couple years.
Great, thanks, Steve, Nuno. Jeff, what challenges or opportunities do individual differences between subjects introduce? Individual C. elegans are likely to be very similar, but for other organisms, other species, there are going to be differences. How do we account for that?
Well, this is a great question, and I participated in that study I showed of the 8 connectomes, and to our surprise, 40% of the neuron-to-neuron connections, even in C. elegans, were different from one animal to another, and these were isogenic animals. So I think variability in wiring diagrams is something you're going to have to deal with no matter what species you look at. Maybe the most interesting thing is that variability has many sources, and some of them we don't understand why they exist. Why would isogenic animals have different wiring diagrams to some degree? One answer to that question might be that some of their wiring diagram is instantiated based on the individual experiences of that animal. We don't think of that very much when we think of worms, but it really hasn't been ruled out. But when we think about mammal brains and all vertebrate brains, where a good deal of learning is stably maintained for the life of the animal, it's a reasonable hypothesis that that information is going to be instantiated in the wiring diagram, and therefore different animals will have different wiring diagrams. And the challenge here is how to harness this variability to learn something as opposed to seeing it as an obstacle, because the differences, which always are asked, and whenever anyone talks to me about connectomics, one of the first questions is, how can you make sense of something if everyone is a little different from every other one? I think learning how those differences get into the wiring diagram and what their purpose is, where that variability comes from, is one of the fundamental challenges. And one of the ways to do that is looking at a lot of animals. And I should say, just going back to this question of expense, there are huge expenses for infrastructure of building wiring diagrams. But once the machines are there, using them for another wiring diagram is very, very inexpensive. So once the infrastructure exists, one can ask many questions. And I think one of the kinds of questions one would like to ask is, how does that region of the brain look from one animal to another? Animals that have, for example, very similar age, gender, genetic line, but may differ only in, for example, their experiences. Thanks.
Thanks, Jeff. Our next question is to Eva from Carolina Teguatl. Apologies for the pronunciation. And she asks about the capabilities of machine learning to help in this field, how we can use machine learning to do segmentation and identify different aspects of the large neuroimaging data sets. So things like cell types or regions of the brain. Kind of where are we with using computers to help to make sense of all of this data?
Yeah, it's a great question. I mean, I think in general, a lot of the progress that we've seen has been, tracking much of what's happening in machine learning, which is, kind of exploding in terms of applications across the. Typically, we have to provide these systems with training data, right? And training data, as we've kind of heard from many of the presenters and even, discussions of proofreading, right? Being able to collect the amount of training data required to often, detect these different objects. Of course, when you're, looking for different types of objects, now you have to retrain a network to potentially be able to find these different types of structures, right? And so I think we are making tons of progress in being able to solve these segmentation challenges using, at this point, large amounts of training data and also large amounts of proofreading efforts afterwards. However, I think, as we're continuing to track the progress of ML, I think that there's a lot of really exciting advances that are happening there in unsupervised and self-supervised learning. And I believe that as we're continuing to move to a place where we can leverage previously collected data sets and previously collected annotations and not have to sort of start from scratch each time we want to find a new type of object, I think that we're going to see that the capabilities of these data sets will also kind of explode exponentially as we don't need as much training data and we can actually solve these hard transfer learning problems. So I think we're in a position now where all of this is really ripe for this next generation of discovery in neuroanatomy and in brain connectomics and structure.
Thank you. Kristen, we're probably going to get into a number of questions regarding structure and function and functional data sets. We'll start with this, the dynamics of synapses. How can they be studied using the techniques that we've talked about today?
Thanks very much for the question. Can you hear me? Yes.
Yep.
I think my mute is off, right? You can hear me, right?
Yep.
Okay, good. Yeah, so I think this is a fantastic question, and it's one that I get every time I give a talk. And I think one of the only ways that you can use electron microscopy to study dynamics of function is to do what we call time series 3D EM. So basically, you do comparisons between animals that have undergone different treatments and look at how structure differs under those conditions. Or you look at brain slices. Even if in the same brain, you can give cells and synapses different experiences and compare them, which we do routinely. The other piece that's becoming much more standard now, and that is to do correlative light microscopy, where you follow the life history of an individual synapse and then begin to look at its ultrastructural characteristics following a particular treatment like glutamate uncaging, but then to go to the EM level to learn what structure-function relationships were occurring at the synapse that underwent a change in the size of its dendritic spine, for example, and follow that up with organelle-like analysis at the electron microscopic level. The other strategy, of course, is to begin to label particular synapses that you can expose with light or channel rhodopsin type labeling to an experience and be able to compare synapses and do this sort of sparse labeling and compare synapses even on the same dendrite that have undergone activation to neighboring ones that haven't at least recently undergone the same activation. And this is another strategy we and others are employing to identify synapses that have undergone particular changes in the context of the circuit surrounding them. So I think all of these strategies, as these microscopy tools improve, all of these strategies are going to give us the opportunity to investigate dynamics of the synapses in pretty well-defined states over time. So I think going down this road of getting better tools for the full mouse connectome and applying them in these other contexts is going to give us a much broader view of how structure and function are related throughout the brain.
Thanks, Kristen, very much. We have a question for Steve from Ming Zhen, and his question is, moving toward Petascale and Exascale Connectomics, one of the things that's really important is to develop relevant standards for the data, the metadata, and computational models, as well as the experimental protocols so that both the primary and secondary data analyses can be well understood and completed. And so wondered what your thoughts were on this and how we might move toward that as a field.
Thanks. Fantastic question, Ming. I think this deserves probably hours of conversation, so I'll try to answer it in a minute, though. We have, I think, several challenges across the pipelines And I think each stage represents a certain amount of risk. And the problem that we have when we go to PETA and Exascale is, of course, everything gets multiplied. And so this question is obviously very important, because anything that we have that's not precise is going to be magnified at those scales, and it probably requires a level of, frankly, industrial standards that we typically don't do in lab. A few examples of this just across the workflow: data acquisition, the preparation of samples. It seems like each person kind of has their own kind of magic recipe for doing things, and it actually adds a lot of risk. I mean, the sad case of a couple of years ago, we lost a very key person in the field in this, which actually, holds back, it hurts the community obviously as well. That's one case. In terms of the data itself, there's been conversations about how we best store the data. There have been conferences that have existed, but that is something where the standardization must be better. I mean, even having platforms like, existed for GitHub, for software has been very instrumental in allowing people to get involved and to contribute. So having those types of platforms is very, very important. In terms of proofreading and how we actually analyze the data itself, having a certain set of standards. Again, when you have hundreds of people that are going to be looking at the data, they have to follow a certain way of proofreading the data so that you can ensure quality. And maybe last but not least, the segmentation. You would think in some ways this would be easy, because we have output that we care about, which is a segmented volume that we can proofread, and we have input, which are these different algorithms, and you want to match and see how good the result is. But there's only been a handful of challenge data sets that have been out there, and those challenge data sets that exist only focus on narrow measures that don't actually measure, as far as I can tell, the speed of proofreading, how much will impact the cost of doing connectomics. And so it's very important that all this stuff really gets honed in and honed down and made it better if we hope to do this project going forward.
Steve, thanks so much. Just want to make a quick note for everyone. We are past the 1 o'clock time, but we will continue with some additional questions. We're getting a lot that are still streaming in. Next one goes to Nuno. So Dr. Eve Martyr mentioned yesterday in her talk that transcriptomics alone was not enough for identifying cell types. What role can this community play in this pursuit?
I think this community can play at least two important roles. One of them is mapping the connectivity of different cells, and from there itself, from the connectivity, create a classification of cell types. And we have seen that now in this large data set, that every time we pick a particular cell type, we see some very specific patterns of connectivity. But the other important role is to link that pattern of connectivity and that cell classification that emerges from the connectivity to the transcriptomics, classifications that exist, because the connectivity is basically the structure configuration of the circuit, but the transcriptomic carries kind of the genes carry the instructions about to build this connectivity. And one step that we think it's easier to do first is through the morphology of the neurons, like I mentioned in my talk, using, for example, PET-seq. But I'm pretty sure that in the future will be many other techniques that will allow us to go even more deep into this relationship.
Great. Thanks very much. Our next question is back to Jeff. Sort of a short question with perhaps a big answer, which is, what do we do with the connectome once we have it?
That is a good question. My initial reaction would be like, what couldn't you do with a connectome once you had it? I think we can get some sense of what connectomes are by thinking about data like genomes. What would you do with the human genome once you had it? And you realize that a data set is not the answer to a question. It's the dictionary or encyclopedia you would use to answer any one of an infinite number of questions. And so I think the hope is that if one had a whole animal connectome, there are an uncountable number of different kinds of questions one can ask. Ones such as what do the projections, where do the projections to a particular part of the brain come from? which particular cells are they making synapses on, and what do those cells do with that information? Where do they send it to other parts of the brain? So one would be able to map out the pathways between brain areas at a synaptic level. That would be enormously valuable. But what's not lost on me is that some of the most interesting uses of genomic information have been on things that I don't think anyone really imagined at the time the genomes were developed. For example, the origins of humans have emerged by looking at the relative similarities of DNA of ancestral animals and how humans have migrated across the planet. I don't think anyone thought genomics was going to play a key role there, but it has been transformative for that field. And I have a feeling that connectomics would have the same kind of opportunity, that there are questions that we can't yet pose because the data doesn't exist. But once we have connectomes, my suspicion is there will be a huge number of new questions asked and new answers that the organizers of generating this kind of data could not have imagined at the time they made the data available. Thanks.
Thanks, Jeff. So, Kristen, specific question for your talk. How close can the actuator get back to its original position for subsequent images? How much alignment is required between two images taken at the same position?
Right. So these are all questions that we are continuously experimenting with in our new technology. But the short sort of generalized answer right now is that centering the region of interest is routinely within 2 micrometers on the XY stage at a 45-degree stage tilt. Any changes, of course, in working distance seem to be much less than that. The beauty of this orientation is that we only have to create one dynamic focal plane, as opposed to, if you do tilt tomography, as opposed to one tilt and a cone around that tilt. So that really saves us a lot. And when we go from section to section, remember we're going to be having 350 nanometers or 300 nanometer section thickness, so then we want to connect the next 300 nanometer section thickness. It's the same level, it's the same alignment tool that you use for serial sections, because they are serial sections, they're just thicker. So it's very simple. In fact, it's very simple to align the 2 adjacent virtual sections between 2 serial sections that are that thick. In fact, it's going to be simpler because as people don't often recognize, each section that you take with a diamond knife loses on the order of 7 nanometers of section thickness. And so with a 45 nanometer section thickness, you're losing a much greater fraction of the section. You don't really lose any objects at 7 nanometers. You can follow them easily across that. But the fraction of the overall section that's lost at the knife edge is much less for these virtual sections computed from a 300 nanometer section thickness then versus a 45 nanometer. So I think overall we're going to find that alignment and reproducibility of the location is going to be the least of our problems. The most of the problems are going to be the same as everybody else. Figuring out how to collect and store a large amount of data in a routine fashion. And I think that's something that's rapidly being resolved by our 2 leaders here and many of the others in this group. Thanks.
Okay, so we're running a little tight on time. There's still a lot of great questions that we'd like to get to, but we in this particular forum. But we want to just quickly thank our panel and the audience for their great questions. This is an exciting and growing research space. There's still a lot of questions and ideas to discuss, and we will have a forum in the networking lounge at 2:30 p.m. It's the chat session for Connectomics, so we all invite you to join us there and continue this discussion and to continue with questions and sharing of ideas. And again, thank you to everyone for joining.